details. I will keep on time. This is why I have my watch. So, what is new about vascular and hemodynamic aging? As you all know, I mentioned that yesterday too, before the process of atherosclerosis, we have this process of arteriosclerosis. And this is important because that is one way to find risk subjects early on, before the development of atherosclerosis. But please remember, everything about early vascular aging is about morphology. Changes in the arterial wall, stiffness, less elastin, more collagen, so and so, morphology. Then we should consider the physiological hemodynamic changes, the consequences. On the one hand, morphology, what I show you here. In the other hand, hemodynamic changes. I come back on this. This slide was shown also yesterday. I will not go into the details. This is a summary what I mean by discussing early vascular aging. Lots of changes in morphology in the vascular wall. However, also with some aspects influencing endothelial function and thereby also hemodynamic function later on. I will also present to you some new data from a collaborative project I am running together with colleagues in Moscow. I was in Moscow a few days ago to speak at another meeting, the Russian Hypertension Society, and I will share with you some new, still unpublished data from Moscow, because that is a high-risk population, as you may well understand. Pulse wave velocity is a marker, a measure of arterial stiffness, and as was shown yesterday, especially in the younger subjects, in middle-aged subjects, there is the increased risk associated with increased pulse wave velocity. The interesting thing is that the older we get, the more we resemble each other in a biological meaning if we survive. However, in mid-age, genetic factors, maybe also some lifestyle factors, will impact on the rate of aging. And that is why we will see diversity, heterogeneity. Some people will have a more premature aging profile in biology, in endocrinology, in metabolism, also in the vasculature. Yes, other people for other reasons may have a retarded, more successful aging. This is the interaction between genes and environment, and sometimes just by chance. Where you are born, during the circumstances, birth cohort effects are important. This is a part of Europe with a very complicated history. You know it much more than I do. And during some episodes of this history, it was very difficult for pregnant women to get enough to eat, the nutrients. They lived under stressful conditions, and that impacted on the offspring, the biology of offspring, and increased cardiovascular risk due to early life programming. During other periods, more favorable conditions, better food, more nutrients, and less risk, because that is all a reflection of when people are born during the circumstances. Family history and genetics, just a few new things about it. I mentioned yesterday that if we consider the risk of increased pulse wave velocity, as indicated here, in patients with early onset coronary artery disease below the age of 55 years, this is increased as compared to the controls. But please notice also in the relatives of these patients, we see the increased risk of arterial stiffness. So this is an indication that there must be some genetic factors. Maybe also subject to the interplay with the environmental factors, epigenetics, so and so. But my argument comes now. This is the genetic map of hypertension and blood pressure regulation. You've seen it before, it's interesting, but it's only a map. It doesn't tell you very much about function, it's just a genetic map. But please notice now, because this is my argument. On chromosome 13, you see nothing. No genetic markers of hypertension or blood pressure. However, in another study from Sardinia, Italy, with a control group in the United States, it was shown that exactly on chromosome 13, there is a genetic marker for arterial stiffness. So this means that in spite of the fact that blood pressure, hypertension, and arterial stiffness are closely linked for many, many reasons, there is also an independent component of arterial stiffness, because this is an independent genetic marker. So the argument is, if we can understand this and the mechanism linked to this genetic marker, receptors, mechanism so-and-so, this could be a drug target, a new drug target, to influence that mechanism. On the one hand, we try to lower blood pressure, fine, but on the other hand, to find new ways to provide new drug targets. This is the meaning of the new cardiovascular genetics, not to predict in individuals. It's not that strong, but to find the accurate, true, new biological mechanism, because then we will see drug targets. Coming now to hemodynamic aging. A few months ago, I had the pleasure to publish a joint review with some of my colleagues all across Europe. Professor Kotzi sitting here, one of the co-authors, thank you very much. Other people from other countries, a review on early vascular aging. If you want a PDF, send me an email, I'll send the PDF to you. Because it's a summary, it's an update on EVA and everything about it. So, in this process, there is a crosstalk between the large arteries, stiffness, and the small arteries, and all these hemodynamic changes. So it led me to think about hemodynamic aging as the physiological consequence of the morphological changes. So what do we think about blood pressure changes with aging? Of course, there are well-known age-related changes in brachial blood pressure, isolated systolic hypertension, yes. Elevated pulse pressure, yes. Also in the central circulation, we see similar changes with aging. In young people, as you might know, the central blood pressure is lower than the brachial blood pressure. But with aging, it increases. In Malmö, we do family studies to investigate central hemodynamics in young subjects from families that increased cardiovascular risk and hypertension. So the hypothesis is they have early changes, early increase in the central blood pressure. Now comes the interesting part, because there are other hemodynamic changes, all associated with increased cardiovascular risk for various reasons. But behind them is a common denominator, arterial stiffness. So increased blood pressure variability, visit-to-visit variability, linked to stiffness. Decreased heart rate variability, linked to stiffness, impaired endothelial function, and very interestingly, impaired baroreflexor function, orthostatic hypertension. We know that in young subjects, vasovagal reactions are not dangerous. It's just a problem. But in mid-age subjects and in elderly subjects, in patients with long-standing diabetes, orthostatic reactions are dangerous. Not because you can faint, but these are predictors of future cardiovascular events. Why? Because impaired baroreceptor function is the consequence of arterial stiffness and superimposed atherosclerosis, less baroreceptor sensitivity, leading to orthostatic reactions. So, if we have patients with orthostatic reaction patterns, this is an indication of stiffness, and that is exactly why it predicts future cardiovascular events. So if we consider EVA and morphology, these are the hemodynamic consequences. In modern epidemiology, we should not only look for risk factors, disease progression, so and so. This is a bit old-fashioned. We should look for anomalies. For example, that I discussed this breakfast. Why do not all obese subjects have hypertension? Why? Even if we investigate the full hemodynamic problems, we will find that some of these subjects have normal tension. This is anomaly. If we can understand that and the genetic background of that, there will be new drug targets. You understand? So the old-fashioned way is to look for risk and disease, blah, blah, blah. The new idea is to look for genetics linked to anomalies. In Sweden, we have a project, prolonged study, looking for patients with type 1 diabetes of long duration, 30, 40, 50 years, escaping complications, anomaly, to find the protective genes, to understand the mechanism, and there will be new drug targets. You see the point? So, EVA and HIS, two sides of the same coin, morphology and pathophysiology. Gianfranco Parati, professor and friend from Milan, published this paper, and it was shown that less arterial compliance, this means more stiffness, is linked to increased blood pressure variability, not only visit to visit, but also short. That is one example. Reduced heart variability is another example I told you about. And in this study, it was shown that in patients with type 1 diabetes, impaired lower heart rate variability is associated with increased stiffness. In the Rotterdam study, population-based, it was shown that orthostatic reactions link to stiffness, as I mentioned to you. So the one thing is a marker of the other thing. The same goes for this Greek study. Increased stiffness, the more pronounced orthostatic reaction, hemodynamic aging. Now I move to telomeres. I guess you know about telomeres at the end of the DNA helix, shortening with every cell division. It has been shown that in patients with early onset myocardial infarction, they have shorter telomeres corresponding to a difference between groups of 11.3 years chronological age, meaning that patients with early onset AMI have more advanced aging, cardiovascular aging. It's difficult to measure telomeres, many methodological problems, but there is one interesting genetic argument. This is a map of genetic markers linked to telomere biology. I will not go into the details, but please notice that if you combine these genetic markers together, they predict coronary artery disease risk. So this is an argument to show us that even if we don't understand telomere biology, even if we cannot measure it accurately enough, there must be something. So I told you about my collaboration with my Russian colleagues in Moscow. They invited now over 300 subjects, no medication but increased cardiovascular risk, men and women like this. Later on, randomized to different drug treatments. But please consider only the upper part. So they were invited. They measured insulin sensitivity, glucose metabolism, also leukocyte telomere length, and as shown here, there was an association between leukocyte telomere length, with age of course, but also with markers of glucose metabolism. This is to say that people with shorter telomeres have impaired glucose metabolism and insulin resistance. This is interesting. Please notice, when they compared patients with type 2 diabetes or without type 2 diabetes with the long telomeres, there was no increase in pulse rate velocity, as was shown in the subjects with short telomeres. So in patients with type 2 diabetes, short telomeres, early aging, they have pronounced arterial stiffness. So it goes together. Stiffness, biological aging, type 2 diabetes as a model. They could also show that when they compared subjects with short telomeres and with the long telomeres, There were differences according to pulse wave velocity, intermediate thickness, and plaque prevalence, atherosclerotic plaques. This is not pulse pressure, plaque prevalence number. So, markers of vascular aging. In Russia, in Moscow, in a high-risk population, it has been shown like this. Finally, new drugs for vascular protection. I mentioned to you yesterday that people are trying to find new ways. There is something called compound 21. It's a specific angiotensin 2-82 agonist to press the button, the 82 receptor, with all the beneficial effects for vascular protection. Not blood pressure lowering, but vascular protection. As shown in this animal model, when they treated rats with LNMA to increase blood pressure and pulse wave velocity, when they added compound 21, this new drug, blood pressure was not very much reduced. However, pulse wave velocity, stiffness was reduced. And later this year, it is hoped that this drug will move from animal studies to human studies. This will be extremely interesting. Maybe it can be combined with resveratrol, as you mentioned, or some other things, or with blood pressure lowering drugs, but this is for vascular protection. We will see. So in summary, I told you about EVA, about hemodynamic aging, some new ideas, and the new drugs. But the final remark is that we have to change our thinking about biology. We should try to understand biology, but not only look for risk and disease, but to look for the anomalies. For example, obese patients with normal blood pressure. Why? You see? Thank you.
Thank you a lot, Peter, for this very elegant talk. Are there any questions?
Congratulations for the excellent lecture that we can learn something new. I would just like to ask you, this is research, what are the practical applications? Because there is a big overlap between lymphocyte and terminal lymph. So practically we can, let's say you said it's difficult, but we can measure and get those results in our patients. How that would be useful in this stage of research?
Well, to be honest with you, telomere length should not be applied in the clinic today. But we can ask very simple questions about family history, about previous life experience. And as trained clinicians, we watch our patients. Some look accurate to their age. Some other people look prematurely aged. We wonder why. So some very simple things can be combined with more sophisticated things. And I really hope that measurement of arterial stiffness also during 24 hours will be much more used in the future, because central hemodynamics, arterial stiffness will bring much more information as compared to the simple brachial blood pressure. This is a 100-year-old mistake. Sorry, protocol, 100 years old mistake, because the brachial blood pressure is not what is important. The central blood pressure, for many reasons, is the important one.
Okay, second question. You know that many groups, even from the Atlas study, introduced edema media fitness of the carotid arteries, and they then said there is a chronologic age, chronologic age, and there is a vascular age. And there is a new concept incorporated in the risk score charts that you also can calculate vascular age. So how do you think how that incorporates in your test?
I have a critical thinking. Of course, it's good if you try to find like a risk algorithm for vascular age. But previously, this was only done based on some conventional risk factors, like Framingham did. Conventional risk factors calculating vascular age. Intermediate thickness is not good enough. It also takes ultrasound. But to measure arterial stiffness could be done in a much better way. So I think we should move on, not only to rely upon traditional risk factors, but to measure what really means something. Stiffness.
One more question. Actually, we still have one more question and then we'll go.
Sorry, sorry, Peter. Congratulations on the talk. Just a very brief one. The study included diabetics, and the telomere length has been shown to have great importance in the diabetic population. Is there any difference compared to the others? Because there are many things that might influence vascular aging or accelerate diabetes.
I think I have to ask my colleagues in Moscow because this is still unpublished and they are analyzing the data. But Nebojsa, you are very right. We should focus much more on the type 2 diabetes patients and what's going on. So I will come back on this.
Okay, thank you.